Hi, my name's Mia Case, and I'll be discussing and analyzing the paper Absolute Quantification of Specific Proteins and Complex Mixtures Using Isoto-Coded Affinity Tags. And so it's published in 2004 by Liu et al. And this paper introduces the VICAT technique, which is a strategy for quantifying low abundance proteins in complex matrices. I'll walk you through the methods, how it compares to traditional techniques, and what future directions we see this going in. And so the first thing I want to start off with is what is the importance of quantifying proteins? And so this is really important in modern proteomics. We need to measure how much of a specific protein is present in a system or sample for applications such as biomarker discovery, disease diagnostics in the medical realm, drug efficacy, biological mechanisms, and just other systems in biology. And so traditional quantifications like Western blotting, which I have a schema of how that works right here, is dependent on antibodies, antibody quality, its semi-quantitative nature, and it just has a lack of reproducibility. And so Liu et al. Emphasizes this chemical labeling called VICAT, which overcomes these drawbacks and especially for biological matrices that are more complex and antibodies may not bind as efficiently or proteins are low in abundance. And so VICAT enables precise and reproducible quantification using mass spec, and it offers a clear advantage for both research and potentially clinical settings. And so, what are VICAT reagents? And so, this method uses three distinct chemical reagents, and this is a light, heavy, and VICAT and marker VICAT. And so, each reagent is designed with a structure that targets cysteine-containing peptides. The VICAT light, is used to label sample-derived peptides. That is right here. Then we have the VICAT heavy, which is used to label internal standard peptides and includes different isotopes for differentiation in mass spec, which is right here. And then we have the VICAT marker, which is labeled with a radioactive carbon 14 for gel visualization. And so all these reagents contain a biotin group for affinity purification, a photocleavable linker, um, and an isotope-coated region. And so this combination enables for purification and quantification, which is the total goal. And unlike ICAT, which is isotope-coated affinity tagging, um, ICAT lacks this visual feature, which VICAT has, and this improves specificity and tracking, and it allows researchers to isolate proteins or isolate peptides and measure peptides with high precision. And so, what is the objective of this study? So, the primary objective is to develop a workflow using VICAT reagents for quantification of specific proteins. So, the authors selected human group 5-phospholipase A2, which we're going to call HGV, as a model protein, and then we spiked it with two different complex matrices. And so, there was an SF9 insect cell lysate and a murine macrophage lysate. And so the study aimed to show that VICAT could achieve high sensitivity and it would be very reproducible and we could quantify low abundance proteins. And so the specific goal included one, synthesizing the VICAT reagents, which we just talked about, optimizing reduction and labeling protocols, using isoelectric focusing for separation, which is focusing peptides and separating them by their charge, and then validating this method with liquid chromatography, mass spec, and using selected reaction monitoring. So a key aspect of the study was comparing VICAT-based quantification against Western blotting to assess the reliability and just that it is better than the traditional methods. And so this experiment and research contained a very cohesive workflow design. And so, first, the proteins are reduced to expose the cysteine tags. They are then labeled using VICAT light or heavy. Then the labeled proteins are digested with trypsin to reduce or to produce peptides. And then a radioactive VICAT marker is added to act as a tracking reference in the isoelectric focusing. So it's just so we can keep track of that's the visual component. And so this separates them by their charge, which I had previously mentioned, and then the biotin labeled peptides are captured on a streptavidin coated streptavidin-coated beads, and then a photocleavage is used to release these purified peptides. And then these peptides are analyzed using micro liquid chromatography coupled to an electrospray ionization and ion or trap mass spec, specifically using the SRM mode for the quantitative analysis. And each step in this process enhances the specificity and reduces background noise and enables us to identify or quantify low-level proteins. And so the isoelectric focusing plays a crucial role in the workflow. And so the VICAT marker peptide, which is radio-labeled with carbon-14, allows researchers to visually detect and locate this region in the gel where the target peptide migrates in the isoelectric focusing. And so this is essential for extracting the correct gel slice before affinity capture, and the researchers confirmed the reliability of the gel tracking system by comparing it with a radioactive signal intensities from X-ray film and scintillation counting, which I guess consistently co-localized with the expected peptide position. And so it not only this focusing not only reduces the sample complexity, but enhances the mass spec sensitivity by eliminating irrelevant peptides. And this is an example of isoelectric focusing by charge as we go from pH 10 to pH 3. And so we have the affinity capture and photocleavage. And so once the correct gel band is excised, the biotinylated peptides within it are enriched using the streptavidin-coated magnetic beads, and the strength of uh, these coated bead interactions ensures that VICAT labeled peptides are the ones that are retained and not the unlabeled ones. And so this prepares it for mass spec because the peptides are then released from the beads using this ultraviolet um, or this UV light, which cleaves the photocleavable linker in the VICAT reagent. And this is a clean chemical release process and avoids harsh solvents, enzymes that could degrade the peptides, and just reduces overall variability in the samples. And so the resulting purified peptides are ideal for the liquid chromatography mass spec analysis because it has low background interference and it has high analytical sensitivity. And so for the instrumentation, the mass spec that was used was the Thermo LCQ Deca XP ion trap instrument, which was equipped with electrospray ionization. And I have a schema of that. Um, this is where it uses the SRM mode. And then here's the electrospray ionization. And so the researchers use the SRM technique because it tracks specific peptide transitions from precursor to fragment ions. And this allowed them to distinguish between the light and heavy labeled peptides based on their mass. And then they were able to calculate the abundance given those signals. And since the internal standard, which is the heavy, and the experimental, which is the light, differ by only their isotopic composition, the MS response is directly comparable. And the use of micro liquid chromatography enhanced the solution and minimized sample loss, which is critical when we're working with these low abundance peptides. And so for the quantitative results and the Western blot comparison, the VICAT method demonstrated strong analytical performance when the HGV was spiked into the SF9 lysates at different concentrations. The authors or the researchers saw a clear and linear relationship between these two, and that is denoted right here. It had an R value of like almost 1, so it had a high correlation coefficient, and the lower limit of the quantification was 50 femmoles, which is very low, and that showcases the sensitivity of this method. The chromatograms right here showed tight coelution of light and heavy labeled peptides, and the signal to noise ratio exceeded 200. So that was also very good and showcased more of the sensitivity. The results validated the accuracy, the precision, the reproducibility across a varying range of concentrations. And in addition to this, the liquid chromatography mass spec data, the authors compared VICAT quantification to Western blotting in the macrophage lysates, which is right here. And so we can see that the Western blot detection of HGV provided very unreliable and weak nonspecific signals, even with two different antibodies. And so in contrast, we have the VICAT, which provided strong quantifiable mass spec signals for these same samples, which is very different compared to the two signals, which is really strong compared to really weak and lots of background noise. And so this comparison highlighted VICAT's performance with detecting low abundance proteins in cases where antibody based approaches clearly fail. And so VICAT has its strengths, limitations, and so for the strengths, it offers advantages. It's accurate, absolute quantification without relying on antibodies. The use of the isotope coding and an internal standard allows for precise comparisons between different samples, the biotin enrichment, and the isoelectric focusing purification increase the specificity. The photocleavage ensures clean peptide recovery without any interference. The method is highly sensitive, and the quantification is very low for a limit of detection. And additionally, the VICAT marker facilitates this gel tracking feature, which is missing in um, other chemical tagging strategies, and such as the Western blot. And together, these features make VICAT a very excellent tool for validating low abundance proteins, and it targets complex matrices, which is also really important. But for its limitations, it can only label peptides that contain cysteine, and this excludes a large portion of the proteome, which is not very good. It uses the use of the carbon-14 isotope raises concerns about safety and disposal concerns. The multi-step workflow is very complex. It uses gel separation, UV cleavage. It's time consuming and it's not ideal for high throughput um, environments. It requires specialized equipment. It's costly. It can only do one peptide at a time, which limits the scalability, especially in if this were to be used in a medical setting. The reagents are also just complex, and there's many steps to this method. And this hinders the widespread adoption of this technique. And so, given these limitations, for some proposed follow-up experiments would be to adapt VICAT for quantification of phosphorylated peptides, which would be a lot easier and would probably encompass a little more of the proteome. And so this could be achieved in enriching phosphopeptides and then the chemically modifying them to introduce cysteines, which would also expand this method. And this could be then labeled with the VICAT reagents that we talked about and quantified using the same instrumentation, such as the mass spec based approach. And this strategy would enable quantification of signaling pathways and signaling pathway components involved in disease diagnostics, biomarkers, and other biological systems. And so synthesizing non-radioactive fluorescence would kind of alleviate some of those safety concerns, and also this would make it a little more accessible to other facilities that lack infrastructure with this instrumentation and just resources. And so, in conclusion, VICAT is a very robust method for quantification of proteins at low abundance. It integrates chemical labeling, affinity enrichment, visual tracking, um, mass spec detection with a very cohesive workflow. Um, but while without its drawbacks, VICAT represents a very significant advancement over traditional antibody-based methods such as the Western blotting, which we compared to, and untargeted mass spec methods. It has precision, reproducibility, adaptability, and it validates proteins at low abundance. And so future versions could enhance these strengths and benefits and try to target compatibility and support this in a higher throughput environment.